Now let's have a look at the user interface. On the first page of our website here, I will load the user interface by clicking right there. So before I start, everything I will describe in this video and the followings can be found in the documentation tab here. So let's have a look at the dataset as a whole first. So I can decide to color the cells by, say, first cluster. And I validate this choice by clicking down there, show UMAP. That will load the UMAP. So you can see here a representation of every cell in space based on their gene expression. So as you can see, the cells have formed groups that we identified and we named them clusters. So each of these clusters represents a cell type. We found 211 of them, and each of them represents a different cell type. So let's have a look at a few of them. In order to load the legend, the color legend of this annotation, I'm going to select a few clusters. To do so, I do a long click here on the UMAP and make a box. Then I double click the box and that will load the zoom. This zoomed in region now show the legend of the annotation of the cells. For instance, this blue cluster here was annotated as ASCII and feed neurons. By double clicking on the UMAP, I can reset the UMAP to this original zoomed out full version. And if I give you as another example, a zoomed in of this crowded, densely populated region in the center here, you will see that again, the legend does not appear. And that is because our user interface cannot compute more than 20 annotation at a time. So if you say you're interested in this cluster here, you can zoom it even more by zooming a second time in a second box. I double click and I load now the zoomed in window that show the annotation. This cluster was hypodermis cell type 2. So I'll double click on the UMAP to reset it. And now I can choose, for instance, in the color cell by the second option, time point, load it by clicking show UMAP. Now you can see that the color has changed because the cells are annotated by their age. So I'm going to show you one cluster, say this one here. So now that I click and zoomed in this cluster, you can see that in this cluster, we have cells of different ages, day 1, day 3, etc. And if you go down to the second UMAP below, you will find the same thing that I've shown you earlier, the annotation of the UMAP. So this is an AUA neuron, and we have cells of different ages. Now that you know how to use the UMAP visualization, let's query a gene. So we can, for instance, find where it is expressed. So I'm going to use the same tab, Aging Worm Atlas UMAP here. Now I'm going to pick the option Gene Denoise. And now that I pick Gene Denoise, the Select Gene box is available, and I can enter by clicking on it the gene of interest. For example, let's imagine I'm working on the gene called COL2, which is collagen type 2. And I click on the gene. If you do not see any option or any gene when you enter the gene, that will mean we did not detect a single read for that gene in our dataset. I'm clicking showing UMAP to load the expression UMAP. And now you see that the cells are colored by gene expression. So it is this gene expression is expressed in log of the normalized expression. And here you see that only this cluster express call2. So I'm going to select it, zoom in, and so you see that the cells that express log2 are here, and if I look at the UMAP below, you will now see the annotation of the cluster. So this cluster corresponds to the green cluster. However, you see that there's a lot of other cells that are kind of contaminating this UMAP. So what I'm going to do is I'm going to zoom in even more to reduce the number of cell type in the window, and then now I have less cell types and there's no more ambiguity. This cluster is VD motor neurons. So call2 is expressed in VD motor neurons. I double click to zoom out, and now I'm going to query a second gene, say the transcription factor DAV16. I enter it, it is there, I click show UMAP. You can see that this gene is expressed more broadly. Now I'm going to show you this tool here. Say I want to highlight the clusters where DAV16 is expressed less. So to do so, I can use value max here. I look at the value of my expression of all the cells, and I decide to select log1 as a value max. I enter 1, show UMAP. So now every cells that have an expression above log1 are maxed out in dark blue, and you see that even the cells that express lower at lower levels are also darker, so that helps me to identify and see them. I can go even lower by entering 0, and now you see that the cells become even darker, so I can clearly see which cell expressed or not. Inversely, if I enter a value min, say I enter 1, log 1, now you see that all the cells that have an expression below log 1 are grayed, and only the cells that have an expression above log 1 are shown. So that allows me to find the clusters where DAV16 is the more highly expressed. So here I've used the option for called gene denoise. I'm going to reset those. And this really is using a corrected expression that imputes missing values to some cells with lower expression. These values were obtained using a SCVI package. So it helps to visualize the expression more easily with less heterogeneity. However, you can verify the raw expression by clicking raw expression and loading the same type of expression UMAP. And you will see now that you see the raw data, the raw expression of this gene. What you will find is that there's usually a good match between denoised expression and raw expression. However, it is easier to visualize the denoised one. So what I usually prefer to start by working with the denoised expression, and then verify the expression by clicking raw and then looking if the raw match, or not the expression.